Do you still have your grandma and see her around? If so, you are very lucky. But isn't it sad to see your grandma getting older and forgetting things? Hello everyone, my name is MD Mahmudul Hasan and I am a postdoctoral fellow in the School of Computer Science and Engineering at UNSW. But today I am going to talk about my PhD topic, which is about looking at neurodegeneration through the eyes AI-based diagnosis. I worked under the supervision of Professor Eric Mehring, Professor Arkor Somia, and Professor Michael Karoniatis. Well, let's get back to the story of my grandma. My grandma got kind of neurodegenerative disease such as dementia. The problem is she never wanna see a doctor. In fact, she had seen a doctor, but she never wanna see her again. She complains a lot. Doctors give a lot of tests. The tests are costly, time consuming, blah blah blah. I thought for a while. Well, the only thing she does routinely is eye test. In fact, she loves to see the optometrist regularly. Then I searched online. But what I have found is terrific. Neurodegeneration is one of the main reasons of death among Australians. The problem is there is no total cure for this disease, but early diagnosis is very important because early diagnosis can only save the lives. I searched more. Then I have found that there are certain changes in human eye, especially in the retina, due to the neurodegenerative diseases like dementia. Bingo, I got my PhD topic and started working on it. In my PhD, I am working on developing kind of computer-aided tool for the diagnosis of neurodegenerative diseases like dementia, Parkinson's or Huntington's disease using AI or artificial intelligence. Well, I searched more for my research purpose. Then what I have found is that neurodegeneration can be of two types. One is retinal neurodegeneration, like glaucoma, and another is cognitive neurodegeneration, like dementia or Parkinson's disease. So my another study was to look at the differential diagnosis between retinal and cognitive neurodegeneration. Well, let's look what retina looks like. So this is a human eye, what you can see. Behind the human eye in the background, at the back part we see the retina. And this retina consists of different layers, which is called retinal nerve fiber layers. So this is the place where human eye meets the brain, where all the neurons connects the eye from the brain. So I especially worked with the retinal nerve fiber layer thickness values and also worked with ganglion cells, which are the cells that comes from the human brain. So you can always say retina is the window towards the brain. Let's talk about the features that I have used in my study. In machine learning or artificial intelligence, features means the attributes or the properties that you use from a specific thing. Here, the features that we have used is retinal nerve fiber layer. So, if this is the optic nerve where human brain meets the eye, you can consider it like superior in the upper region, inferior in the bottom region, then temporal near the ear, and nasal near the nose. And then it is divided into 12 clock hours. So all this thickness, so actually if you take the retinal nerve fiber thickness values, if you stress this out, it looks like this, which is called RNFL thickness, retinal nerve fiber layer thickness. In my machine learning based algorithm, I have used those thickness values for my analysis. What I did, I plotted those retinal nerve fiber thickness values for healthy patients and for healthy subjects and the patients. And then I applied a kind of mathematical transformation which is called fast Fourier transform. This transformation is used to convert the spatial domain features to frequency domain. And then we get some new features which are really interesting. So these frequency domain features, in addition to the spatial domain features, we have used in the machine learning models to train the models and to understand how the disease looks like. Another important thing is explainable AI that we have used in our study. Look, nowadays you can see the great hypes of AI. People are talking about this everywhere, but most of the people don't want to trust this, especially the clinicians, because they can see this kind of black box, but they don't see what is happening inside the box. So one of the main contributions from my PhD was to unbox the black box so that clinicians can trust the machine learning model to be used for the diagnosis purpose. Well, so one of the concept that we have used in our experiment is sharp or sharply analysis, which considers the features like players in the level playing field. And consider there are 11 players in a soccer or football game, and if there is a goal, that is not actually the contribution of a single player but also contribution of the other players. So it considers the contribution of every single player to the diagnosis of the disease, in my perspective. And the explanation is using partial dependency analysis, where it considers every features partially, it excludes other features, only including one features, and then performs the analysis, and again is it excludes the other features, and only using the single feature it performs the analysis. Thus we understand the partial dependency of each of the feature in the model. We also use this analysis. Using this explainable analysis, what we did, we fed through the features, and when those features went through this explainable analysis, it ranks the feature based on the Shapley values. And here you can see it ranks the most top feature as RNFL symmetry, RNFL inferior thickness. So when the RNFL symmetry is lower, then there is higher chances of being diagnosed with dementia. In this case, we only worked with glaucoma for the first study, so higher chance of glaucoma. On the other hand, for the higher value of RNFL symmetry, higher chances of being diagnosed as healthy patients, healthy subject or normal. If the RNFL thickness value is lower, then the higher chance of being diagnosed with glaucoma. On the other hand, if RNFL inferior thickness value is lower, is higher, then higher chance of being diagnosed with normal subject or healthy. We can also plot those in dependency plot or a scatter plot, and you can see a clear separation, like in when the RNFL symmetry value is less than 75%, then there is higher chance of being diagnosed with glaucoma, otherwise normal. We can also show the dependency between two features. For example, when the RNFL symmetry value is RNFL symmetry value is lower, and also when the RNFL inferior thickness values is lower, then higher chances of being diagnosed with glaucoma, otherwise normal. So what we did, we used those explainable features in the analysis, and we created two apps. The first app is uh, an Excel app where the clinicians can put different thickness values or different features, and in the output it shows the chances of being diagnosed with glaucoma, and it shows the sensitivity of 89.3% and specificity of 87.9%. We created another app, which is explainable app, where we are using the explainable features, and if the uh, clinicians would put those from the mobile phone or from any web explorer, then they can find that if they put the feature values, they can identify what is the chances of being diagnosed with glaucoma or not. If glaucoma, then the contribution of each of the features towards the prediction. Now then what we did, we gave the same data to the clinicians and the machine, and what we found that um, machine learning based models performance, AI based models performance is better than humans. For example, here this is the performance curve of machine, and this is the performance of the human clinician expert. On the other hand, for the different stages of glaucoma diagnosis, you can see for early, moderate, and advanced glaucoma diagnosis, the machine performance is higher than the human or clinician performance. Overall, our machine learning based model, which is random forest based model, outperformed the human clinicians in early stage and overall glaucoma diagnosis with 10.4 to 12.6% higher accuracy. Well, a second study focused on differential diagnosis, where we differentiate between the neurodegeneration, which is optic neuropathy, versus normal or healthy patients. Then, if it is diagnosed with neuropathy, then we identify whether this is glaucoma, which is retinal neurodegeneration, or non-glaucoma, which is cognitive neurodegeneration. If cognitive neurodegeneration, then the chances of being diagnosed with glaucoma, Parkinson's, or ischemic optic neuropathy. We also did explainable analysis over there, and we created this web app where clinicians can put their features. And once they put their feature values, it gives the output, which shows the percentage of being diagnosed with optic neuropathy or normal. If optic neuropathy, it shows the whether this is glaucoma or non-glaucoma. And it also shows the contribution of different features. Here, this explanation is global explanation, so the features that we identified, and the explanations that we get during test time is called local explanation, which shows the contribution of the different features throughout the time. So this helps the clinicians to go back to their system and check the features which contributes to the diagnosis of the disease, rather than checking hundreds of features from their system, which takes a lot of time. Well, then we give the same data to the clinician and to the machine, and it shows that machine outperforms clinicians in the diagnosis. So, uh, overall, our machine learning based model SVM outperforms clinicians with 26.3% higher accuracy than the human clinician or human experts. Look, then we did our third experiment, which is on prediction of the visual field from OCT. So, what visual field is? OCT, what visual field is? It is a functional measure. So, where the patients need to click on some stimuli, and for the older patients and patients with dementia, they could not do it. There is a problem for them, and some clinical facilities also don't have that machine. So we use the structural measures, that is the thickness values from OCT, to predict visual field, and we predicted using the thickness values, we predicted the local measures and the global measures of visual field. We created an explainable app where the clinicians can put different values and predict the outputs over there. Our fourth study was a combination of all those the structural and functional measures, where we put OCT scans, which is optical coherence tomography, color fundus photographs, and visual field functional measures together to fuse, and we created a model, and we compared our model with the state of the art models. So our model performed the multi-attention based multimodal fusion network outperformed the state of the art models. We also compared our multimodal model with the unimodalities, and it performs the best, which you can show from the bar plots. If you want to explore more, you can look at our experimental papers in scientific reports, and also the literature review in clinical and experimental optometry. Well, let's get back to my gamma. I discussed and shared the findings of my PhD with my grandma, and now she's so excited because she's going to see the optometrist and she's going to share the findings of my PhD with her. Don't forget about your grandma. Check up with her and ask her as well. Thank you.